Today we are going to discuss nucleic acids and biological function. Nucleic acids and biological function. Nucleic acids, like I said, are polymers of nucleotides. They are what? Polymers. You don't have polymers of what? Nucleotides. And nucleotides are heterogeneous molecules consisting of three components. The first component is phosphate group, the second one is sugar, the third one is nitrogenous bases. And the sugar can either be ribose in the case of RNA or deoxyribose in the case of DNA. The nitrogenous bases are also categorized into two broad classes. The first one is purine, the second one is pyrimidine. Okay, and the purines are further broken down into two. We have the adenine and we have the guanine. And also the pyrimidines are broken down into three subgroups. We have the uracil, we have the cytosine, we have the thiamine. And I would have you to know that in RNA you have two pyrimidines and two purines, which include the adenine, the guanine, the cytosine, and the uracil. But in DNA, there is a little permutation whereby you have the what? The adenine, the guanine, the cytosine, and the fourth one, the thiamine. You don't know it. So nucleotide looks like this trinity. You have a phosphate group, you have a sugar group, and you have a base. Okay? So you are going to get the phosphate, the sugar. It's always in that tandem. The phosphate, the sugar, the phosphate, the sugar. Till eternity, it does not change. But the third one there is what? The base. And the bases are shielded inside. And they keep changing. The order of the bases represents the order of the genetic information. And an alteration in the order of that sequence is what is known as what? Mutation. So in a great detail, we'll be looking at this concept. And I told us also that one nucleotide is joined to another nucleotide via a what? A bridge. And that bridge is called what? Phosphodiester bond or linkage. I'll take it again. The nucleotides are joined together through a bond, a linkage, a bridge that is called what? Phosphodiester bond. Look at my demonstration. Phosphodiester bond. That means that the phosphate is the parent atom. But to the right or to the head, there is one ester. To the leg, there is another ester. So that you have phosphodiester bond. Are we together? The backbone on each strand consists of what? repeating unit of sugar phosphate group, repeating unit of sugar phosphate, sugar, till the last nucleotide. Okay? And the rungs, R-U-N-G-S, rungs. You know what is rungs? The rungs are made of what? The bases, which can either be purine and what? Pyrimidine, linked together via what? Hydrogen bonds. Now, God is wonderful. The hydrogen bond there can either be dihydrogen bond or trihydrogen bond. The rule is that if adenine pairs with thiamine, you are going to get 2 hydrogen bonds. And if guanine pairs with cytosine, that is the rule, we are coming to that, you are going to get 3 hydrogen bonds. So where you have 2 hydrogen bonds and where you have 3 hydrogen bonds, they are not the same. Okay? The sugar phosphate bond is single bond. So you have single bond, double bond, triple bond, single bond. I will say it again. You have single bond, double bond, triple bond, and what? Single bond. That is how the maker made it. And you will agree with me that it is easier to break single bond. True or false? It's also a lot easier or more difficult to break double bonds. And it's a whole lot of difficulty to break triple bonds. Therefore, if you have DNA sequence that is rich in guanine cytosine ratio, it is very stable and requires high amount of energy to hydrolyze it. Are we together? To break it down. If you have DNA, another DNA that is rich in 80, 80 means adenine thiamine, is easier to break such DNA molecule than it is when you have GC. Are we together? Let's go to the next slide. So what we have there is a typical structure of oligonucleotide. My grammar has changed though. I didn't say nucleotide. I said what? Oligonucleotide. You now have oligo. Oligo is a Greek word meaning few. Few. So how many sugars are there? 3 sugars. You can see sugar number 1, sugar number 2, sugar number 3. Everything there is sugar. Can you see it? And that sugar is pentose, isn't it? All right, now, so the topmost sugar there has a phosphate group. I would have you to know that phosphate is PO4 minus, isn't it? P is phosphorus. O is what? Oxygen. The minus there means it has lost some electron. All right. Then the next one there is at 3 prime. Okay? Is at 3 prime. I would have you to know that the atoms of the sugar are numbered with prime to distinguish them from the atoms of the nitrogenous bases. Are we together? That is beautiful. And the order is usually 5 prime to 3 prime. 5 prime to 3 prime. You can see the 5 up. The next one will be 3. The next one will be 5. The next one will be 3. Where you see 5, you see 3. Where you see 3, you see 5. In that alternating order. Is that clear? Now, the letters that are painted there, are the bases. The first one there is what? Cytosine, C. The second one is what? Adenine, A. And the third one is what? Guanine, G. Now, I want you to understand what I mean by phosphodiester bond. So it suggests there are presence of two ester bonds. The first ester bond is 5, look, don't look at me, is 5 prime. Can you see it? 5 prime, then you have carbon atom. Then what we call ester is carbon oxygen bond, isn't it? Carbon oxygen bond is what we call ester. The first one is coming from 5, or if we change it the other way, we have the first one is coming from 3 prime. Okay, I'm reading from up, the first 3 up. You have, everywhere you have a bend, an angle is carbon. Okay, so if that is correct, let's start counting from O, oxygen atom on top of that sugar structure. Are we together? To the right, you have the first carbon. Take away your eyes from the cytosine. Oh, I'm not talking about the first angle there that is not having C in it is carbon atom, and we call it anomeric carbon. We call it what? Again? Now, this anomeric carbon usually determines the functional activity of the compound. So, and it can either be alpha or beta. Okay, I'm taking you through it can either be alpha configuration or beta configuration. Are we together? It is alpha configuration if the hydrogen atom is up, is in the place of that C. You see C there. If that hydrogen down is up and the C is down, it is alpha. Hello? Are you with me? Now, it is beta configuration if it is like this. If it is like this. If OH is up and H is down. If OH is up and H is down. In this case, our H is down, isn't it? So it is beta configuration. For it to be a sugar, that place is supposed to be OH, isn't it? But that OH has reacted with cytosine to be able to have cytosine there. So it's no longer OH, but what? C. But originally, it was OH. So if OH is up and H is down, it is beta configuration. Therefore, the sugar configuration we have in DNA structure is what? Is beta configuration. Beta D glycosidic linkage. Is what? Beta D glycosidic linkage. D there means dextrorotatory. It rotates. By the time you shine light to it, it will turn the light, in a clockwise direction. So let's continue our nomenclature there now. The second curve there that has H up, H down is carbon number 2. And if you look well, you see that there is no OH here. There is no O in that carbon number 2. Because of that, this structure is deoxy. Deoxy. If I must call it in full, it is 2-deoxy, to tell you that oxygen is missing at carbon number 2. Are we together? Yes, sir. Then we now move to the third angle there. That is carbon number 3. And we move up to the fourth angle there. That is carbon number 4. And then there is an arrow pointing up and is having methyl group, CH2 group. That CH2 group is the carbon number 5. So with these few points of mine, I've been able to convince you that this structure is what? 2-deoxyribose. 2-deoxyribose. 2-deoxyribose sugar. So we have the first ester bond there, which is located at, if I'm coming down, which is located at 3 prime, and we have the second one at 5 prime, where you have CO is the first ester, and where you have CO or OC. If you are coming down, it is CO. That is first ester. And you descend again, you have what? OC. And then phosphate group is in the center. That is called phosphodiester bond. Okay? And that is what I try to explain in that structure. There are 2 types of nucleic acids. Who know? We have deoxyribonucleic acid and we have ribonucleic acid. Deoxyribonucleic acid is DNA. It has a double structure, double strand, not vertically placed, but in a helical version, like a snake, like a serpent in Genesis chapter 3 verse 1. And then we have that DNA carries the genetic information. So any day you hear genetic information, it's just telling you A, T, G, C. The way they are seated is the information. Okay? It's just like if I ask you to spell Godwin, it already entered your brain. You can see it in your mind, isn't it? If you mix the letters, if you swap the letters, it's no longer Godwin. Okay? So the order is called the information. That information could be ear, it could be teeth, it could be lip, it could be neck, it could be any part of your body. Are we together? Yes, sir. And so it carries the genetic information. Very important. It carries the genetic information. Very, very important. Now, it suggests that if DNA carries the genetic information, then it is not the whole structure of DNA that is the information. I want it to sink. That DNA carries what? Genetic information. It therefore suggests that it is not the whole sequence in DNA that is the information. I want to use you as an example. Your body carries your nose. Where does your body start? From your head. Okay. So keep going. Keep going. Keep going now. To your leg. Okay. And you'll find your nose somewhere in between your eyes and your mouth, isn't it? So that means this side is not the genetic information. And anywhere before the information, we call it the upstream. Biochemistry. You're welcome. Anywhere before, any nucleotide before the information, where the information is located, is called the what? Upstream. And anywhere after the genetic information is called what? The downstream. Been to a body of water, you will always identify where the current is coming from, isn't it? Where the source is coming from, is flowing. Okay? Anywhere before where you are standing is up, and anywhere after where you are standing is called the what? The down. Okay? So that means within a stretch of DNA, we have pockets of genes. In human genome, all the genes in human being is about how many? 3.2 billion base pairs. Many of you have not seen billion before. But the genes, which are regions that code for proteins, are just 23,000 genes. In other words, so when they say DNA carries the genetic information, you should understand now. Not that DNA is the genetic information. It carries it. It carries it. Okay? It also implies, as I'm teaching you, I'm getting inspiration. It also implies that there are more non-coding regions of the DNA than we have the coding regions. The genes are the coding region. They code for protein, they code for neck, they code for eyes color, they code for hair color, they code for anything. Are we together? But the greater portion of the DNA do not code for anything. So we have more non-coding region than the coding region. Very important. And somebody may ask, what are the functions of the non-coding region? That is where you and I come in, to explore those non-coding regions. So all the stories I told you yesterday about technology, it happened in the coding region, which is the genetic information. So we have greater unexplored encoded proteins, and so the genetic information code for protein, code for RNA. That means the mother of RNA is what? DNA. The mother of protein is what? DNA. Are we together? So DNA is the father. So the proteins and the RNAs, they mediate the biological, physiological actions of the organisms. In other words, they are the ones responsible for the actions, for the functions of the organism. Okay? All the things you do now is because of the proteins that are translated from this DNA. So DNA is a fascinating molecule. I mean, may I also tell you, all you cannot do now and in the future is because you don't have that gene that can be translated to what you can do. You cannot talk like me because you don't have the gene that can talk like me. You can't look like me because you don't have the gene that. But you can actually get it from me if you desire it. I taught her yesterday. You guys missed yesterday. So, including transportation, enzymatic signaling, regulations, all of them is because they have been translated, from the gene. So all you cannot do. And I also told her yesterday, look at what is the structure of DNA. What is the structure? How does it look like? Because we need to know how it looks like to be able to make something out of it. How does DNA look like? Two names that you see there, James Watson and Francis Crick, they never did any work on DNA, but they were smart people.